Please, Sandra, can you start?
Yes, thank you, Juliana. Well, hello everybody. Welcome to this session on the climate change impacts on shallow lakes. As you know, this is the third day that we are running this session. It was also run by Erik, Jeppesen and Tom Davidson on Monday and Tuesday. So this is the third and last day. Well, first of all, I would like to thank very much the organizers of the Shallow Lake Conference because it's going everything very smooth and we are having very nice time watching all the presentations. And I also would like to thank the presenters of today because I really enjoy watching your presentations. And well, sorry, my name is Sandra Brucet. I work at the University of Vic in Spain, okay, and I will be sharing this session. And well, many of you already participated in the previous session, so you know how this works. You should have watched the videos of the presentations. And now what I will do is to present a very short summary of your presentations, and then we will go for questions. To present this summary, I have picked up some slides of your presentations, okay, just that the people remember the content of your presentations. And well, so now I will share you this, let's see. Can you hear me well, by the way? Because
yeah.
Yes.
Okay.
Yes. Yes.
So let's see. Can you see my presentation now? So here there are
Yes.
the corresponding authors of the presentation and the titles. Okay. So I will follow the order of the agenda. So please, the ones that are the corresponding authors, have your videos on so that the people can see you when they are asking you questions, okay? So the first presenter is Maria Spoiler, and the title of the presentation is Plankton Diversity and Traits in Shallow Water Bodies of Temperate and Mediterranean Regions of Europe. And here, they compare the zooplankton community in water bodies of temperate and Mediterranean regions in order to provide insight into the function of shallow lakes in warmer climate. So this is a space for time substitution approach. They show that the zooplankton community in temperate region is mainly influenced by macrophytes, whereas in the Mediterranean it's influenced by conductivity and fish, both negatively related to zooplankton diversity. They also show that large cladocerans are more abundant at temperate climates and at lower trophic state, whereas small cladocerans were more abundant in the Mediterranean climate and at higher trophic level. They also found that rotifera were more abundant at higher trophic levels in both countries, and copepoda in the Mediterranean were abundant with increased macrophyte abundance. So these results are interesting because they suggest that warming may induce changes in the zooplankton communities, such as the changes in the relative abundances of large and small sizes. So now I stop sharing the presentation, and where is Maria?
Here I am.
I'm here.
Hello,
Yeah.
Sandra. Hello, everyone.
Hi, how are you? So I invite you now for questions to Maria and colleagues.
Hi, do you hear me?
Yeah.
Can I make a question?
Okay. Yeah,
okay,
yes.
Thank you. Hi, my name is Rafael. I would like to ask you if your approach is actually similar to what we know as the space for time approach. You know, instead of, if you want to predict what will happen to these communities in the future, with predicting, if you expect an increase in temperature in the future, can we use this data about different temperatures in space to try to predict what will happen in the future. And based on this, how do you think acclimation may affect the conclusions?
Yes, it is a very, thank you, Rafael, for your question. And yes, this is one point that which we didn't calculate it in that, but yes, probably it should be in the future take in account, but now we make that modeling and prediction according to our data set, what we have and what we can gather actually in very heterogeneous groups. You know, it is not easy to collect data actually spread around Europe, and it is very big differences in the morphometry of the, actually some of those lakes are lakes, some of those are ponds, and also, yes, but I understand. Excuse me? Yes, but I think, yes, okay, thank you for your opinion, and it should be valuable taken into account in the future modeling.
Okay, thank you.
Are there more questions? You can simply speak or raise your hand if you like. I have a question for Maria, if there are no other questions. You said that the conductivity was very important variable in the Mediterranean, okay, which does not surprise myself because it's normally quite important. But the ranges of conductivities were similar in the Mediterranean and the temperate lakes?
Actually, ranges didn't show significant differences, but anyway, conductivity in Mediterranean lakes, especially in that Albanian, which are mostly lagoons, are quite high in 45 microsiemens practically, and yes, and from other side, we have also higher electroconductivity in that northern lakes because of trophic state and because of such nutrients level, and because of that we have such discrepancy actually, but they didn't result in significant differences at the end. But each of these lakes actually have conductivity of different reasons. Different bases, as you can presume.
Thank you. Because we have done some studies also here in Mediterranean ponds and shallow lakes, and conductivity, when you include a lake that has some conductivity, even if, I mean, even if it's oligoline, for example, the conductivity starts influencing a lot the communities. When you go to higher conductivities than fresh water, let's say.
I hope we will solve and explain better all these multi-level questions in our article, hopefully.
May I just make a question? The changes in salinity are mostly due to the change in conductivity are mostly due to changes in salinity.
In Mediterranean lakes, that is definitely connected with salinity. Definitely.
Okay, thank you. Now I see that Pablo has put in the chat the link where you can find the PowerPoint presentations, the PDF, right? The PDF of this session.
Yeah, that's true. It's only the PDF and only from some of the presentations.
Mm-hmm. Okay, if there are no more questions, we'll go to the second one. Fine. The second presentation is by, sorry, I show you the slides. Okay, the second presentation is by Raquel Neves, and the title is Effects of Heavy Rainfall Events on Water Quality Indicators of a Urban Multi-Impacted Lagoon. And here they show how extreme rainfall events influence the abiotic and biotic indicators of the water quality in a very nice urban lagoon in Rio de Janeiro. And the results show that after extreme rainfall events, there were significant changes in phytoplankton abundance, which decreased, and Escherichia coli, which increased. So these results show that extreme events are very important for the communities and the functioning of these lagoons because they can lead to changes in the phytoplankton community and the water quality. Okay, Raquel, I cannot see her. Raquel is not here. Actually, I did not receive her PDF. So it seems that she's not here. Is there any colleague from her? No. Okay, so there is no one that can ask questions. So I don't know if someone has questions, but they will not be answered. It seems. Okay, let's go to the third presentation then. Okay. Okay, the third presenter, I hope she's here. Eva, are you here? Otherwise, we have a problem. Or any colleague of Eva? Because the next presentation is from the same group.
Colleague,
okay.
But yeah, I'm a student, so I don't know if I can answer the questions to her presentation. Okay. I'm quite not competent.
But do you know if Eva will connect, or do you know something?
I just asked her, but I didn't.
Because maybe we can wait. So,
yeah,
so let's do something. Let's do something. We go to your presentation, which is the next one. Okay.
Okay. I'll try to do my best.
Presentation for the last one. Okay. So let's go to yours. Okay. This one. Okay, this one. So the title of your presentation, Wojtek, sorry, I cannot pronounce your surname. Wilczynski, can you pronounce it, please, for me?
Yeah, it's Wilczynski. Actually, it's quite hard.
Wilczynski. Okay, thank you. So the title is the combined effect of an elevated temperature and the presence of hypoxia on the interspecific competition of Daphnia. Okay. And here the authors assess the mechanisms behind the decrease of body size of zooplankton with increased temperature. They test if larger body size zooplankton are less competitive compared to the small ones because of the oxygen limitations associated to higher temperatures. So they perform experiments at low and high oxygen concentrations at 2 temperatures and with 2 pairs of zooplankton species which differ in size. And the experimental results show that the temperature size pattern is only confirmed in one species combination, Daphnia magna versus Daphnia pulex. And in the case of Daphnia longispina versus Daphnia pulex, the hypothesis was not confirmed since larger body sizes competitively excluded a smaller species. So they concluded that, contrary to their hypothesis, low oxygen concentrations may sometimes favor larger body size species, possibly due to higher hemoglobin concentrations in their tissues. And they also conclude that there may be other mechanisms involved in the temporal size pattern besides oxygen and temperature. Okay, so I invite questions for Wojtek.
I do have one question.
Yes, please.
Okay. So I was wondering if this temperature and oxygen may act like a predator in terms of, for example, cascade effects. If you, for example, in terms of if the large plant increases under these conditions, will it lead to, for example, higher grazing and diminish, reducing algae biomass or something like that? You know, if there is some kind of effect on lower troph levels.
Wojtek?
I should start answering. I did not really understand everything. I understood that temperature and oxygen could work together, and temperature could affect the grazing ratio of Daphnia. Yes. So yes, it could. But
sorry, not just the grazing effect of Daphnia, but the grazing effect of the whole troph level. You know, the community effect on the algae, because you're changing the proportion of different species.
Yes, certainly the algae were. I think there's some graphs of the algae concentrations. I could try to share my screen, and I'm on my phone, so I could show you the graphs of the algal concentration changes. They were changing, and they were changing differently in each treatment. They were the highest, the concentrations of algae in our experiments at the beginning of the experiments, because the density of Daphnia was very low. And then they reached a plateau at around 0.2. So the Daphnia were indeed causing an effect on the algae, but I don't know what kind of effect. I would have to dig more into the data, and that was not the point of the experiment. I think I'll contact my supervisor and do some research on that.
No, that's okay. That's okay. Thank you. Thank you so much for your answer.
Thank you.
Any more questions? Let me check, because Mariana says that she cannot, that the microphone is not working.
Hello?
Yes, please go.
No, I'm the next one.
Okay, no, Mariana has a question, and she says, were they lab clones, or did they come from the field? A question for Wojtek.
If the clones we used were cultivated in laboratory, yes, we obtained them in the field and cultivated them in a laboratory for around 2 months until they were fully capable of producing offspring that we used in the experiments. So yeah, they were fresh clones that we cultivated in the laboratory for a short period.
Okay, thank you. Any more questions? Marcus, please.
Yeah, thanks for presenting the study, Wojtek. It was really interesting to watch, I think. It's more a question out of curiosity, like based on your results now, what would be your next step in your research? Like what would be, yeah, the addition to what you observed?
Wait. I have trouble with this Zoom. Save driving mode. What is? No. Okay. Sorry. We were planning on testing the hemoglobin concentrations in the tissues of Daphnia. Can you hear me?
Yeah, we can hear you very well.
Sorry, I'm having trouble with Zoom. So mainly I'm studying code and the chemical warfare agents, but as far as I know, my supervisor is planning on testing the hemoglobin concentrations in the tissues of our tested Daphnia. We froze the specimens and we are planning on analyzing them in a molecular way to find the cause of these effects that we have observed.
Thanks. Yeah.
Okay, any more questions? Otherwise, we go to the next speaker. I will share again my screen. Okay, next speaker is Mariano Bresciani. And the title of the presentation is Analysis of Time Series of the Essential Climate Variables from ESA CCLX, a case study in a shallow Italian eutrophic lake. And here they use the high temporal resolution of the CCA satellite data coupled with the RS5 climatic data to study Lake Trasimeno in Italy, shallow turbic and eutrophic lake. They show that this data is very valuable to understanding the interannual dynamics driving phytoplankton blooms. And it also allowed them to classify lake ecological status to moderate and poor, and to assess the time periods and areas that were critical for the water framework directive evaluation. So please, I invite you for questions to Mariana.
I had one, if can go.
Yeah, Mariana.
Thank you, Mariano. It was a really cool presentation and data set, and especially you have these latest years where warming or extreme heat wave in the summer has been shown up to discuss. And I was wondering, so how did you ended up relating annual TP with September chlorophyll A concentration? I was just curious, maybe I understood it wrong, but you relate the annual average of TP with one specific month of chlorophyll A, which is September, and I was curious, so if you could evolve more about this decision.
Okay, yes, this is a game, more or less, that we have tested. We have selected September because it's the period with a higher concentration of chlorophyll A, more or less in all years of the data set. And normally in Lake Trasimeno in September, we have a bloom of cyanobacteria. And we have tested if in this period we have this positive regression respect to the phosphorus concentration, because we don't have a clear evidence that the thermal condition highly represent the bloom of cyanobacteria. And we think that another reason could influence this great peak of cyanobacteria in September, and could be the concentration of nutrient. Unfortunately, we have a very useful and long data set with remote sensing data. We have 1,000 of satellite products. We have in situ measurement with respect to the reflectances that provided to us chlorophyll A, total suspended matter concentration, phycosaline pigment concentration, but we don't have a great data set of nutrient inside of the water, because we are expert in remote sensing technologies. And we have only the data of local authority, and we use an average value of phosphorus for the year to evaluate this regression. The results more or less show that when we have the year with higher concentration of chlorophyll A, we have this positive regression with higher concentration of nutrient, but probably is necessary increase the analysis respect to the nutrient inside of the water.
Thanks. No, now it's totally understandable. So you don't have enough data, I guess, for TP. But I was wondering if it was for any other reason. So yeah. Perfect. Thank you.
Welcome.
Any more questions?
Can I make one?
Well, please.
Mariano, thank you for your presentation. Sorry, I wasn't able to see the video, but I would like to ask you one thing. Sometimes when we have this kind of data, we relate the predictors and the responses at the same time. Do you actually thought about analyzing this with some kind of time lag instead of relating the data that you have of temperature or nutrients of each time step instead of relating to the response of that same time step? Wait for some time, wait for months, I don't know, or years to see some kind of time lag effect?
If I understood, because it's not easy to understand, we are inside of my presentation, the aims of my presentation of my work and I want to show to you is that we provide a long time series data set of some parameters obtained from satellite data, okay, in this case is for Lake Trasimeno, that could be very useful to monitoring and to understand the dynamics, for example, of phytoplankton and chlorophyll A in the water, in a shallow lake's water, such as Lake Trasimeno. It's not a prediction, this is a really data, because remote sensing provide true data of some parameters that optically interaction with the light and the concentration obtained from satellite have the same quality of in situ concentration obtained. We have this information and yes, sure, we possible create a data set to understand or think about what's happening in the lakes. Yes, but it's another part of work. It's necessary to have some modelers that put inside of the modeling all the parameters that could be influenced the phytoplankton dynamics. For example, for the cyanobacteria, you know that wind and light intensity is two the parameters that have a very important role in the bloom of cyanobacteria. And the modelers take our data, meteorological and weather data, put all together and create a modeling to predict possible bloom. In Lake Trasimeno is a little bit complicated because the wind condition is very strange. You have a different situation that alter the normality of phenomena in other zone, and for the modelers it's not easy predict the blooms.
Thank you so much.
Thank you very much, Mariano, for the nice presentation. Are there any more questions? Okay, then we go to the next one. Okay, next presenter is Marcus Herrmann, and the title is Facing Future Temperature Extremes and Changes. How do freshwater ecosystems under chemical stress respond to recurrent heat wave and increased temperatures? And here they perform an experiment in indoor mesocosmos to assess the effects of chemical stress and heat waves on zooplankton, macroinvertebrates, and mollusk species, and also some ecosystem processes. This is a novel experiment since it combines the temperature increase, the chemicals, and heat wave, which occur a long time. They observed that, for example, that the heat wave had an effect on dissolved oxygen, which generally decreased during the heat wave. Chlorophyll A was higher in the control compared to the higher temperatures, and also compared to the mesocosmos with chemicals. They also show a significant decline in the abundance of macro and microzooplankton species, such as Daphnia and Chidorus, just after the heat wave and under chemical stress. And in contrast, copepods were much less affected by thermal and chemical stress. For this presentation, there were already some questions in the Moodle. Okay. By who? Bastian Post. I don't know if he's here, otherwise I will ask the questions. Okay, so he says that it's a great talk, a nice experiment, which I agree. And he asked, why did you use a fungicide and not an herbicide targeting the macrophytes plankton, or an insecticide targeting the zooplankton? There are several questions, so maybe you can first answer this one. He's here, Bastian. So.
Yeah, hi, but Marco's gone answering. It's it's fine if you read my question anyway.
Yeah, I also replied to it in this group.
You replied already. Okay, so I didn't see.
Yeah. But maybe just a short catch up, like, because this is a quite common question I got asked actually, like, why did you select a fungicide? Why not a fancy new insecticide or so? And we had several reasons in mind why we choose carbendazim in this study. You can all read the answer, which is mainly about the target biota group, but also in addition to what I wrote in the forum is actually because we know already quite a bit on how the chemical itself acts in the system. So what the physical chemical properties of the compound is. And since we are really interested in multiple stressor effects, we wanted to have a really long interaction time of the thermal stressor on one hand, but then also on the chemical stressor. So of course, if you apply a compound that might be also commonly used in agriculture, but that shows, for instance, a very fast dissipation rate, then your interaction time of the stressors and the biota is relatively short. So the probability that you might actually reveal interaction effects goes towards zero. So yeah, it's a lot about background knowledge to answer this question for the choice, but it's indeed a valid one and a good one.
So the other questions, I understand they were already answered as well.
They were all answered.
Okay.
Sorry.
I have a question. I have a question. Do you have any idea why cyclopod were much less affected than the other species?
Yeah, so the data, the preliminary data that I presented was mainly abundance data, right? So it's basically species count, and there might be several reasons like why certain species were higher counted or less. So one possible answer to your question is that life cycles in there might differ, and that also that the life cycles are affected at a different stage. So yeah, while some of the species we had in the community of 29 species, now I can tell, I mean, while they were rather sexually or parthenogenic reproducing, they show different life cycles to others. So maybe even under these conditions, they benefit from it and can still tolerate it. But I'm open-minded for other explanations, because I'm actually just analyzing it as well. Yeah. Thanks.
Well,
Marcus, I also have a question, if that's okay.
Yeah, sure. Go ahead.
Yeah.
Hi.
Hi.
I really enjoyed your talk, and I'm also working on something similar. I also tried a microcosm experiment, and it's quite hard, also counting out the samples, but it's fun. I was wondering, so you now show the responses of specific species under your treatments. Have you also looked at the total community composition and if it changed under high temperature or not?
Yeah, thanks, Lana, for your feedback. Really appreciate it. And I really enjoyed also your talk about the PCO2. Yeah, that's also a good question. I mean, what I presented was really just the first results out of this. As you can imagine, this counting zooplankton is really time consuming, especially with a high number of replicates, which we had in this study. So yes, the next step is indeed also community analysis of the whole community. Now we know we have identified 29 species in the community. So that's actually the final goal, to apply multivariate statistics and see how the community is responding under the different treatments. Yeah.
And what do you expect? Do you have a hypothesis or no idea of what?
Yeah. So with this experiment, we were also a bit digging in new, kind of new areas in the way that, as it has been already presented by the beginning of this week, that heat waves are relatively under-researched. There's not so much knowledge about it. So what we speculate is indeed that the extreme event in combination, oh, bless you, with the chemical stressor has the largest detrimental effects on the community, so that we can really see a decline. It would be interesting if we even see kind of an adaptation response during the experimental time period, but a valid point in this study is for sure that we didn't include a recovery period. So in another outdoor study that we conducted last year, we also included a recovery period after the treatment, and there we are really curious in how these systems can maybe go back to their initial state or to a next stable state. Yeah,
it's very interesting work. I want to follow your work. Keep me updated.
Yeah, sure.
Thank you.
Yeah. Thanks.
Yeah, I think there is another question.
Yeah, also from my side, thanks a lot for this very nice presentation. I'm collaborator of Bastian, so you might be involved or informed on our work. I was also wondering on the choice of the fungicide, so I don't ask this question again, but what I was interested in, do you have any idea on the modes of action on the fungicide of the zooplankton, because you find mainly effects on that? This was astonishing. My second question, did you include any fungal related endpoints beyond the microbial activity, which was mentioned in your presentation? So a direct effect, or do you have any ideas on indirect effects already?
Yeah, so thanks, Maite. These were, really good questions. Thanks a lot. And to recap the first question about the MOA of carbendazim, I mean, since it is actually an applied fungicide, it's engineered to kill fungi, right? So it's actually inhibiting, I think, like cell reproduction. I think, if I'm not mistaken, don't get me wrong. But the interesting part in that applying the fungicide was indeed that in a previous study, already quite some time ago, also a mesocosm study without temperature, but the same compound 10 years ago. In this research group I'm now in, they figured out these detrimental effects first. And yeah, in our experimental design of this study now, it was also, I mean, of course, a challenge to select the right concentration, because if you have seen in a previous study that the compound is indeed causing adverse effects, causing mortality on zooplankton species, then you need to choose also the right concentrations, right? If you apply it, and we just gave it one shot, so we gave it the 100 micrograms per liter based on the previous observations. And of course, if you go wrong, if the concentration is too high, you will basically kill all and you will not see any interaction. If you go too low, you might not see anything happening compared to the control. So it's really all based on previous knowledge from the research group here in Wageningen. And yeah, so this is what I can say so far about the MOA. I haven't digged deeper. I think you might have expected a bit of a different answer. I don't know, but we can talk about it anytime also with Bastian. What was the second question about again?
It was related to fungal endpoints.
Yeah.
Related to the original mode of action of the compound.
Yeah, so to direct and indirect effects, that's indeed also, as I replied in the forum, what we are currently analyzing actually. So we had in the study 10 different endpoints to assess ecosystem function and processes. And what we would like to do is to really see, okay, if there is a direct relation between these endpoints. Is there direct effects if chlorophyll A is increasing or decreasing? Can we relate that to, for instance, total abundance count or so? Yeah. And we also had another PhD in the group who kind of specialized in structural equation modeling. So of course, then the next step, really nice step, would be to evaluate this data in a more modeling way to see these direct and indirect effects and to see if there are indeed significant yeah, data flows, let's say. Yeah.
Thank you.
Thanks for the question.
Thanks a lot, Marcus. Very nice
Yeah,
presentation.
sure. Thank you.
Any more questions? No, so I share again. So next speaker is Rafael de Toni Guariento. The title is The Temperature Dependence of Animal-Mediated Nutrient Recycling in Aquatic Ecosystems, Reconciling Predation, Physiology, and Stoichiometric Constraints. And here they predicted that aquatic invertebrate consumers will require higher feeding effort and carbon-rich diets in warm environment because of the increased respiratory demands, which in turn would increase nutrient release rate. So they evaluated this prediction using a stoichiometric model. Their prediction was not fully supported because the model showed that the resource requirements of aquatic invertebrates change with temperature in a U-shaped relationship. So the consumer nutrient requirements at first decreases, but then increases with increasing temperature. And it exhibits a minimum at optimal temperatures. They also found that a diet switch to rich food at elevated temperatures, and it compensates for the extra metabolic demands, especially when feeding efforts is restricted due to predation. Sorry. So Rafael, okay.
Yep.
Rafael, you are here. I invite questions to Rafael.
I have one. Ben, you're well done. I never put my hands up, but okay, go.
No problem.
Thank you, Rafael. I wrote down your email because I am really curious to know more about the model, I guess, or how you design the model. But I wanted to bring up here about, because how do you take into account the carbon quality? So from my understanding, you talk about carbon-rich food, but then you play with carbon to N ratios, which to me, if you increase these ratios, doesn't really mean higher carbon quantities. And then it also means less quality or less food quality in theory. So I was curious to know, how did you approach this difference between carbon quality and carbon quantity?
All right. People sadly that my mic is not very good. Actually, they are having trouble to hear me. I don't know if you can hear me, but I will try to do my best.
We can hear you, but not that well. So.
Yeah. Okay. Sorry, guys.
No problem. We can hear you, but we need to concentrate.
Okay. So this actually the goal here was to challenge some previous work, some previous predictions about how effort will scale with temperature. Mostly because the expectation is that effort will increase with temperature, because carbon is already abundant in the foods of these herbivores. So there is enough carbon in the, I don't know, in what they eat and the primary producers, right? So if they want more carbon, they just have to eat more. So increase temperature, increase effort, they will get the carbon that they need, and they will be okay. However, we're trying to impose constraints. And what were those constraints? Predation risk, and also different efficiencies in assimilation efficiency of nutrients along the temperature gradient. And what we saw, what the model gives to us is not is the optimal relationship of effort and carbon acquisition. So when temperature increases, these animals will need more carbon. They will actually eat on lower quality food because they don't need nutrients, they need carbon. And the prediction was the previous work showed that they just had to eat more. However, what we are showing is that when we have constraints to foraging, especially due to the risk of predation or other costs related to increasing effort, they will actually switch diets. They will switch to the lower quality food, to the higher carbon to nitrogen, because this carbon is what they need now, especially due to the threshold elemental ratio function. So they will be limited for carbon instead of nutrients. So they will switch to lower carbon foods because it's easier to get the carbon that they need eating on lower quality food instead of just increasing carbon content. Because to increase the carbon content acquisition, they need to increase effort. And increasing effort, there are some problems related to that, especially because they are more exposed to predators, because there are other costs related to foraging and so on. So instead of increasing quantity, they will switch diets when these constraints, they are present.
Okay, thank you.
Thank you.
So I have another question. Thanks. I'm going to be honest, I haven't watched your talk, but I'm like, this is a very cool topic for me, and I apologize for not watching your talk in advance. You answer most of my questions by answering Maria's, but I have a follow-up. So considering Luke's talk today in the morning, how do you think the role, like Luke the Mister's talk today in the morning about local adaptation and like warming adaptation, thermal adaptation, how do you think thermal adaptation will play a role in these trophic interactions when it comes to, so if currently your model has constraints such as the handling time or the amount of food that they consume or the simulation efficiency, do you expect the thermal adaptation will decrease the impacts of thermal stress and therefore will like kind of level off? And an additional question is, here you're only looking at the effects of temperature on like zooplankton, let's say. What about if you consider the effects of temperature on the phytoplankton stoichiometry?
All right, thank you. I understood both of your questions, and they are really good actually. So the first one regarding the temperature adaptation. Actually, there is a paper by Craig White. I think he was the last author in Nature Climate Change. I think 3 years ago. It's a meta-analysis that he showed. Of course, there is a acute response and a chronic response. And the acute response is higher, of course, and the chronic response is lower. So in the modeling that we did, the effort is free to vary. So the model will give us the best effort. But we can also set other parameters to vary and look for the optimal parameter based on the model optimization. And I can use, for example, the parameters regarding the respiration rate with temperature or the efficiency in the nutrient acquisition with the temperature to look for some compensation, this kind of acclimation effects that these organisms will have with temperature. I did not perform this yet. I still want to do that. So my goal is to let these parameters regarding the efficiency and how respiration increases with temperature to also vary with temperature and look for the optimal values and see how these interact with the effort. So my goal is to compare how effort changes when these parameters are fixed and when these parameters can change. So I can take a look at the effects of thermal adaptation in this whole scenario. And the other question is about the effects of temperature on the algae stoichiometry, on the primary producer stoichiometry, right? I thought that doing that would be too complex. I would be going to all kinds of directions and I may lose my first goal. My goal in this study was to focus on this diet switch and then constraints. But also, this could be very important in case I need to generalize my results and so on and look for, I don't know, things that change together. So if I can have an expectation that this stoichiometry will change together with temperature, maybe this is an important thing to do in the future.
Awesome, thanks. These are amazing answers. I'll watch it and I'll catch up with you later because this is very relevant from our work currently.
Oh, sure. Just send me an email and we can chat more.
Okay, any more questions? Yes, Tobias.
Yeah, I have one. Hi, really interesting presentation. I was thinking about how you modeled the predator-prey relationship. So you say you include predation risk or no predation risk. So did you include actual predation, so removal of prey, or did you see a behavior shift in the prey if you modeled a predator? And you also, in your talk, said you solved this optimal foraging behavior. How did you do that? Could you explain which algorithm you used, or could you just, I'm really interested to see, how did you actually see this shift in behavior? Thanks.
Yeah. And thank you so much for your questions. Okay, I think I understood both of them. So the first one's about how I modeled predator-prey interactions. And I can do both ways. I can actually just look at the behavior shifts. So there will be no effect on the density of the prey because the model is composed of two parts. The first part is optimization modeling, and the second part is a multi-color simulation. In this multi-color simulation, I can remove prey to look for the effects of the prey removal. So I can do both things. But in this presentation, there is just one effect, just the behavioral shift, the effort. change with the predator presence. But I can do both if needed. And how the model works? Actually, this model is a kind of model that is used to understand animal behavior. I think there is two important books in this case, by John McNamara and by, oh my God, I forgot his name. It's a Canadian, Mark, oh sorry, I will look at this. Oh my God. It doesn't matter, I'm not remembering right now under pressure. So I have a paper, actually, just briefly. I have a paper in functional ecology. I think it's 2 years ago, 3 years ago. Just look at variant of functional ecology. And I have a description of the model in the methods. I even have an algorithm description in the methods. And it's basically the same model. It's just a bit more complex. But if you want to take a look at how the modeling, Mark Mengel is the guy that published books about this kind of modeling. And if you want to take a look at more specifically how the model works, I suggest you to just take a look at this paper that I published in functional ecology, and in the methods you have a brief description how the model works. It's optimization modeling for animal behavior.
Thanks.
Could you share the paper link here? Yes, I can share the paper link here. As the presentation goes on, I will send the link here. Okay, sure. Oh, it's here. Yeah. Thank you so much for your question.
Thanks, Rafael. Many thanks.
Thank you.
So we go to the next one. Okay. Wait, sorry. Yeah, next speaker is Cleto Jose Freire Costa Jr. And the presentation is Extreme Drought Increases Benthic Macroinvertebrate Beta Diversity in Semi-arid Reservoirs in Brazil. And here they studied the effects of extreme drought in Brazilian reservoirs. They hypothesized that extreme drought deteriorates reservoir water quality and decreases environmental heterogeneity, reducing the functional and taxonomic diversity, and homogenizing the assemblages of benthic macroinvertebrates. But however, against their prediction, they found that extreme drought increased environmental heterogeneity, and this may be the reason of an increase in beta diversity of macroinvertebrate communities. They also found that drought intensification decreased water quality, but caused no effect on benthic macroinvertebrate taxonomy and functional diversity. Cleto Jose Freire is here.
Well, he's not here, but I am.
Okay,
he vanished somehow, but I'm here for the answers, okay, also for the questions. It's good to see you, Sandra, and everyone that I know here, and also the others. Sorry, didn't want to be excluded.
Yeah, nice to see you, and many people that I have not seen for a long time.
So does anybody have questions? I think Maria has one.
Okay, yeah.
Regina wanted to go first, maybe.
Regina, please.
Hi, Rosenberg.
Hi, Regina.
So I saw the presentation, and one thing that caught my attention was in the results in the final, but it's just a commentary actually, because one of the findings is that the decrease in water quality actually was related with higher beta biodiversity. Mm-hmm. And I was just thinking on a manager perspective, how can you present this discourse? Because this is a thing that was mentioned during my PhD thesis, that we need to be kind of careful when we're presenting data like this, because it actually can be thought as okay, the water quality is degraded, but we do have high biodiversity. So I was just wondering how can we, in these kind of situations, how can we manage to present our results in a way that doesn't lead to this distorted discourse?
Yes, thank you for your comment and also somehow a question. Well, it's a little bit complicated actually. Our results, we did not actually, we are not trying to state that the decrease of water quality doesn't have an impact, for example, in the function of the ecosystems. But we have to pay attention also in the data and how you handle the data. Our data sets actually, they were sampled in two seasons that actually I cannot even say that there were 2 seasons because it didn't rain at all. I mean, we had a dry and an extremely dry period. And then the water quality was already bad before the extremely dry event. So probably the threshold, let's say, to change the structure of the community or to change the richness, the functional richness of the macroinvertebrate community was already reached even after an extreme event. So we have to be careful on how we handle the data sets and how we explain the results and how we explain the situation, the scenario that we are working with. I don't know if I answered your question, but you have to be careful. If you have a decrease in the richness or diversity or functional diversity, or if you don't have any fact, it doesn't mean that it's good or bad for the ecosystem. I think you have to be able to convey the information for the scientific community and also for society in a very precise way in order to avoid these misunderstandings that we generally have depending on the people that are trying to use our results to somehow say that, okay, it's not very bad, the water quality is decreasing, but the diversity is okay. So you just have to be careful on how we convey the information, I guess. Scientific communication is probably how we handle the scientific communication that will probably be the best way to answer your question, I guess.
Maybe I can follow up on this because I think amazing results and presentation, and maybe because of that, maybe even yourself, you have this discussion session that kind of, yeah, you discuss about your results, which I found it really interesting. But maybe I missed it or something, but I was wondering if you could elaborate more on if this increase in diversity would be maybe more because higher poor quality tolerant species, or like if it could be another reason, which, as you said, it's not good or bad. It just, if you observe this kind of.
Well, I just have to be clear that there wasn't an increase in the richness and diversity. There was an increase in beta diversity. I mean, there was a change in species composition with decreasing water quality. The main explanation for that, at least that's what I speculate, let's say, is that, I mean, the reservoirs, they don't dry out exactly in the same way. I mean, the reservoirs, they have morphologies that are not similar. So some of the reservoirs not drying out, like, let's say we had 8 reservoirs in the extremely dry season, in the extremely dry event, there were some reservoirs that were 20% of the maximum water storage. There were some that were 5% of the maximum water storage. So this increased the heterogeneity among the systems. So that's why there was an increase in the beta diversity. There was an increase in the replacement of species because of this condition. I mean, before the extremely dry event, it was already bad, but the systems were more homogenized, let's say. So that's the answer,
point.
I guess, for your question.
Thank you. Yeah.
I have
Rosenberg. Sorry.
Oh, sorry.
I think Rafael has one.
No. How did you calculate this environmental heterogeneity?
Sorry. Sorry, Sandra.
Rafael, please ask. You.
Okay, sorry for. Okay, I will do it. Rosenberg, nice to see you, my friend. I was in my mind, the drought is a big black box because a lot of things are happening, right? Concentration of nutrients are changing, the interactions are changing, refuges are no longer present, and so on and so on and so on. So I was wondering if this functional approach, knowing what traits are changing, would be an avenue to try to decompose this black box. What do you think about that?
Well, I will try to use my experience with this work, specifically with the macroinvertebrates community. We know almost nothing about the macroinvertebrates, at least in the systems that we sampled, and we cannot actually see any differences in the functional structure of the macroinvertebrates community. There were a dominance of chironomids before and after the extreme dry event, let's say, and well, we don't know much to say that these differences, these changes, like you said, in the interactions, these changes in the environmental variables, how this could impact the functional structure, let's say, of the macroinvertebrate assemblages in these reservoirs. But definitely, we have to take into account. I was actually expecting changes in the functional structure of the macroinvertebrate communities, because we observed that in the zooplankton, we observed that in the phytoplankton, the fish, but in the macroinvertebrates, actually, it was very surprising to see no shifts in the functional, but in the structure of the communities. Probably we have to be more careful, I don't know, with the data sets that we are handling, because there were some of the species that we were not able to identify as well. So there are many species that are in the genus level, for example. So maybe we need to somehow refine the identification to try to answer what you have just asked to me.
Thank you.
You're welcome. Thank you for your question as well.
There is a question in the chat that it says, do you believe that temperature change is the main factor for the decrease in the density of macroinvertebrates, or would you have other factors that influence?
Well, I would not expect so in our case, because we were dealing with systems that were sampled within the semi-arid region in Brazil. There were some differences in the precipitation rates between the basins that we sampled, but there were no differences in the temperature. So I would not expect any impact of temperature in the structure and the functioning of the macroinvertebrates communities in our case. But definitely temperature has an effect, but it was not the case in our work.
I cannot recall, how did you calculate the environmental heterogeneity?
Okay, we used environmental variables. I can't remember all of them. I actually wanted to decompose the calculation of heterogeneity. I wanted to partition it in, for example, productivity variables like phosphorus, chlorophyll, and so on and so forth. Those variables that we were expecting to be related to productivity, those variables that were related to, let's say, salinity, and so on and so forth. But we decided to use all of the variables to calculate the environmental heterogeneity. We used Euclidean distance to calculate the environmental heterogeneity within the lakes. It's like when you calculate beta diversity, for example, using Sorensen breakers and other dissimilarity measures. But we use Euclidean. We use the matrix of the environmental variables and then calculated the environmental heterogeneity that is specified, let's say, as the distances to the centroid for each one of the region that we sampled in this reservoir, in this region. I don't know if it was clear.
No, it was not really clear for me. I saw many variables that you assessed, but
well,
really
basically we had two regions, and one of the regions we had 8 reservoirs, and for each one of these regions we calculated the environmental heterogeneity. Like we calculate the beta diversity calculation, but we use the Euclidean distance to calculate the average distances from each one of these reservoirs to the centroid. The centroid would be the basin. And then we would have 8 values, let's say, for environmental heterogeneity for each one of the regions. So we would end up with 16 distant measures that we called, that we named as environmental heterogeneity.
Mm-hmm. Okay, thank you.
You're welcome.
More questions? Seeing that there are no more questions. If so, let's go to the last one, I think. Thank you very much, Rosenberg.
You're welcome. Thank you for everybody's questions.
Okay, here. The last one is Juan Pablo Pacheco, and the title is Small Size Omnivorous Fish Promotes Stronger Effects on Food Webs Than Warming and Eutrophication Experimental Shallow Lakes. So here they perform an interesting experiment on mesocosmos on the role of small omnivorous fish on shallow lake food webs. So they tested the combined effects of small omnivorous fish, temperature, and nutrients on the phytoplankton and periphyton. They found that omnivory by small fish had a strong cascading effects, enhancing phytoplankton dominance and periphyton loss. And this is because the fish present reduce the cladocerans, and so they release the grazing pressure on phytoplankton. Fish also promoted the grazing on periphyton by snails, because these fish could not eat the snails, so they increase their abundances. And in addition, they observed that the treatments with fish have had less zooplankton and macroinvertebrate diversity. And they concluded that the small fish promote more phytoplankton than the worst IPCC warming scenario plus eutrophic conditions. So this is, of course, an important result to take into account for lake management. Okay, Juan Pablo. Okay. So I invite questions to Pablo.
I do have one.
Okay, yeah, Rafael.
Pablo, hi. I was wondering if you use it in this experiment different densities of the fish and different values for temperature. So, like in a surface analysis, varying both variables, or if the temperature is fixed or not. I wasn't sure if you just have one single scenario for temperature, or if you had different values for temperature.
Thank you, Rafael, for your question. I don't know if you can hear me well. It's okay.
Pablo, now we hear you very low. I don't think, well, it's the same for the others, but now it's worse.
Hear me now?
Yeah.
Now it's better.
Now it's better. Okay, I will try to talk as clear as possible. Well, actually, it was a fully factorial design. So we cross conditions of warming and without warming, presence or absence of a small density of omnivorous fish. So we only use one density of omnivorous fish, and nutrient enrichment versus like no nutrient enrichment. So it was like a mesotrophic versus eutrophic conditions. So that was the original design. We couldn't include many different densities of fish because in the experiment design, there were in total 32 different mesocones, and we had no chance like treatments combining at the same time of fish, different densities of fish. So we have the restrictions of the experimental design there. So we just test of the presence of the fish versus the absence of the fish.
No, I understand. I think, of course, I understand these limitations, because I was wondering, it's not a criticism or anything like that, but I was wondering if some, as a reader, and the expectation when you are saying that this thing is stronger than this, then someone can ask, okay, but if you increase the temperature, so maybe the temperature will be stronger than the fish effect, and so on. I was just thinking about the expectation that some reader will have just reading that this is stronger than this, and then you don't have different values for temperature, different densities for the fish. Maybe try to balance these things. I don't know how, but just a small suggestion.
It's a very good question, actually. In our experiment, we simulated densities of fish, but with very low densities of fish compared to natural densities in subtropical ecosystem. So we include only 5 individuals per square meter. That is like in the limit of the unrealistic. So we can expect that if we even increase more than that, the number of fish will have like much stronger effect. But we decide not to do that because we can have some restrictions. For example, like having like a strong effect during the first days and then not having the same process on zooplankton or macroinvertebrate during the rest of the experiment. So it was like combining conditions of the presence of fish, but not too many fish as we can expect in natural ecosystem. So in subtropical, typical is to have like at least 25 per square meter or something like that in mesotrophic to eutrophic conditions. And here we simulated only with 5 individuals per square meter. That is quite low, but still we have like very strong effect with these low fish densities. So yeah.
Relating to the natural abundance actually is a great way to deal with that. So I think it's a great justification. So thank you.
Thank you for your question, Rafael. Rosenberg.
Hi, Juan. It's nice to see you, and congratulations for your very nice presentation. It's probably a silly but somehow important question, I guess. What about, you were talking about subtropical systems in South America. This is expectations from the IPCC scenario that you tried to handle in your experiment. Can you actually say that these increases in the temperature will be taking place in subtropical systems? And can you generalize that for all kinds of subtropical systems in the world, or?
That's a wonderful question. And the quick answer is no, of course, because the effect of the climate change, they might be different depending on the region. But we simulated this IPCC scenario as also a strategy of a space for time substitution. So it's changing the conditions in temperature to analyze, for example, latitudinal gradient that actually exists between ecosystem. In subtropics, for example, is drastically different the composition of the fish community compared to temperate system. So we tried to simulate that. It was not like the study was not focused on the impact of long-term climate change, or it was not only focused on that, but it was mostly on the latitudinal difference that we can have nowadays between subtropics and temperate systems. So we tried to simulate that, combining this higher temperature with the fish absence, that it was we thought that it was the main factor affecting the trophic food webs and the effects on periphyton and phytoplankton. But yes, we cannot expect the same for different climates. Thank you for your question, Rosen. Yeah.
Thank you for your answer.
Yeah.
Sabine, please.
Yeah, thank you for the nice experiment. I was wondering in how far the increase in the fish, in the snail density with the increased fish is like a common response. So is that also known from like other experiments or from any real work? Because also like in temperate lakes, you seem to have lakes where periphyton grazing is dominated by snails, and then you have other systems where for some reason snails are not so abundant. I was always wondering how that, why that is. So yeah, maybe you can explain a bit about that.
Yeah, thank you, Sabine, for your question. It's a very, very good question. And it's a topic that we are discussing right now, because our experimental design it was like all the snails and invertebrates all together. We cannot separate just the effect of the snails from the rest of the macroinvertebrates. What we clearly see, this drastic increase on the snails. So, and it was only in treatment with fish. So we are discussing that there are many mechanisms in which the fish can be like making to increase the snail density and therefore the grazing pressure on periphyton. One of these mechanisms is by releasing of competition by other invertebrates. So other grazers are just disappearing from the experiment. And another thing that might be happening, besides the lack of competition from other grazers, is that the fish are like eliminating all the predators, and especially the predators. And the predators, they can prey on fish and on snail eggs, sorry. So it can be also a way that is releasing the snails from eggs predation from other macroinvertebrates. It's something interesting. We found some other experiment with similar results. We couldn't evidence any effect of the temperature on snails, that it was something that we were expecting to have, but still it was this like very clear link between the fish and snails acting together and increasing the grazing on periphyton by affecting the whole macroinvertebrate community. So our explanation is more or less that, yeah, well, the fish are affecting macroinvertebrates, and then this is releasing from other grazers, and is releasing snails from a direct consumption from other macroinvertebrates. I'm not sure if, did you understand well?
Yeah, thanks. Yeah, I think the snail, the egg story seems more likely to me, because otherwise, if once a snail is a little bigger, you can't imagine of any sort of competition situation. But feeding on eggs, that might be a point. Thank you.
Yeah, well, and some other thing also to add to this answer is that in some experiment, it was seen that the signal in the water of having a predator of a snail egg decreased the reproduction of the snails. So that's also something important. Maybe it's not the direct grazing on the eggs, but it's some other water chemical response or something like that is affecting the snails in the water. So it was something unexpected to have an increase in the snail predation, but yeah, still an interesting result we have. Thank you, Sabine, for your question.
Other questions? Marcos, please.
Yeah, thanks for sharing your outcome, Juan. That was really interesting. And I think it's quite a complex study, to be honest. I think you really built a quite a complex system. So my point of that I'm always questioning, my question point is always this predator in these test systems, because from what I know from literature so far is that most of the test systems are relatively small. So I have the picture in mind that as soon as you add a predator, it feels like it must feel for them like being in heaven with all the food supply and maybe a little bit of temperature increase, which they can easily tolerate maybe to a certain extent. So my question is, even though in your experimental design you separate, you have treatments with fish and then without fish, how can you still disentangle all the other factors that, if we only look, for instance, on the zooplankton effects, right? I mean, zooplankton abundance might be also affected by macrophyte growth. I think you also had them in your test systems, or phytoplankton abundance. So
I'm not fully sure if I understood, but we cross the treatments to being able to separate the effect, and we include the same densities of zooplankton. So it must be like a control factor that. And it was a complex experimental design to be realistic, the most realistic as possible with the littoral conditions of a shallow lake. So we have to include natural plants, but as we expected to lose some plants, we also include artificial plants to standardize the periphyton area to colonize. So yeah, it has some things that make the experiment quite complex, and we couldn't add other factors as just to include the presence of one fish as to see if it's enough as to cause a drastic change on zooplankton or whatever. But still we play with these very low densities of fish. So we could probably expect to, even with lower densities of fish, will have similar effect.
Okay, and I mean, based on your experimental design, was it then still kind of easy and plausible to identify these effect chains, or is it more speculative, let's say? Like
this effect of the fish, you mean?
Yeah, or in general, I think in your graph you showed effects also on zooplankton or other.
Yeah, well, actually we evaluated the effect on many different factors, and the effect of the fish was very clear through the food web on periphyton, phytoplankton, composition, biomass, on macroinvertebrates and zooplankton. So it was like a very consistent response that we have only with the presence of these small densities of fish. So I would expect that, yes, we can say that are very consistent patterns in our experiment and very clear responses in all levels, even in some levels that we were not expecting to have so clear responses, as in diversity of zooplankton and macroinvertebrates. We have these drastic responses. So yeah, I would say that is very, very clear the response we have. We were also expecting to have some extra effect from temperature, and we didn't have that effect of temperature. So it was like the main factor affecting the whole food web there. It was just the presence of the fish, and with a little of help from the nutrient enrichment as well. Yeah.
Okay, thanks a lot and congrats. Yeah.
Thank you very much, Marcus. Yeah.
Pablo, in relation to this that you were saying now, weren't you surprised to not find higher, for example, phytoplankton in the treatments with fish, high temperature and high nutrients? So,
well, actually we separate the temperature effect, and what we have, maybe I can show here on the presentation, just a second.
Because I think you didn't mention much about this, but when checking the figures, maybe what you are going to show now.
No, this was the figure that you show. I don't know if you can see here.
Yeah, well, I didn't mean this one, eh?
Ah, okay, which one?
No, for example, if you go to the number 13, for example.
So plankton diversity.
Yeah, for example, now I cannot remember for this one, for example. If you check the treatment with high temperature, high nutrients, and fish, which one is this one? The third one?
Yeah. Is the red one? You can see here is the ambient and high nutrient is AH, and warm and high nutrient. So we have
even a bit lower, no, than the one with fish.
Well, it was not statistically different. It looks like lower, but it was not a consistent pattern on that. So compared to the treatment only with warm, sorry.
No, that the temperature and nutrients do not play any role at all. I mean,
the nutrients they play
with fish, they do not increase the
the nutrients they play a role. You can see here with high nutrients versus low nutrients with fish conditions, we have like very different biomass, significantly different biomass, and it's the same here when you combine like in warming treatment, with high nutrients versus low nutrients. There is a difference between those. In the presence of fish. So the nutrients are like making a synergistic or increasing the effect of the fish, but we didn't see any effect of temperature. And that was actually a surprise. Maybe it's because of the cumulative effect of the temperature. This experiment, it lasts 2 months, roughly 2 months. So maybe if this experiment ran for a longer period, we would expect to have some other cumulative effects of temperature, but we didn't find any of them, even on the composition of phytoplankton or periphyton. So, and it was like an extreme, that was strange, actually. It was an extreme scenario of climate change with plus 4.5 degrees. So we would expect to have some changes on composition, but it looks like there are others like conditionals, in this case, the fish are affecting much more and shaping the drastic change on biomass and composition, much more than only the combined effect of nutrients enrichment with higher temperature.
Very interesting. Maybe as you said, with a longer time, duration of the experiment, then you could find some temperature effects. But
it was a short experiment because it was during a stay in China. It was a short time, only 6 months in total from the setup to end with the samples counting. And it was in Wuhan, so it was with the coronavirus outbreak in the middle. So it was quite difficult to finish all the experiment and all the things, but we managed in 6 months. So luckily it was not longer, the experiment, it was only 2 months.
I think it's a very interesting experiment that you are lucky now to be able to show the results here.
Exactly.
After 1 year. So you went quite fast. Anyway, I think it was a very nice experiment, even though it could be longer, but we all know that it's very difficult to run this experiment for a long time.
And it's a part of many different experiments on my PhD, so it couldn't last too long either because of that. So but I think we have interesting things to discuss, at least.
Thanks a lot.
Thank you very much, Sandra, and it was very nice to see all of you around.
Are there more questions? And Ewa is here. No, I did not see her name, so we missed to present one of the presentations, but Okay, I think that she's not here. Okay, so thank you very much to all the presenters and all the people in this session. It was really interesting and nice to see you here, and very interesting discussions as well. So thanks a lot for being here.
Thank you very much, Sandra.
Thank you very much, Sandra.
Thank you.
See you.
Thank you.
Thank you.
Bye.
Bye-bye.
Bye.
Bye.